Imagine you are an oncologist. You have a patient sitting across from you, a 44-year-old man, and he's dealing with a highly aggressive cancer of the bile ducts.
Right, something we call stage 4 cholangiocarcinoma.
Exactly.
Yeah.
So you run the sequencing, and you know exactly what is driving his tumor. It's a microscopic protein switch inside his cells that is just, permanently jammed in the on position.
Yeah, a mutation in the KRAS gene.
Yeah.
Which is incredibly notoriously difficult.
Right. And here is the really frustrating part for you as the doctor. For about 40 years, biochemists have looked at that active jammed switch and told doctors that structurally, it's a perfectly smooth sphere.
Yeah, no pockets, no grooves.
Absolutely nowhere for a drug molecule to latch onto. It was considered chemically impossible to drug. So, what do you do?
Historically, you really didn't have many options. You gave them standard systemic chemotherapy, maybe you tried immunotherapy, but the biology of this specific mutation is just relentless. For decades, having a mutant KRAS-driven cancer, especially outside of the lungs, was essentially a dead end. But, what we are looking at today changes that entire narrative. It's a total paradigm shift.
Which is exactly why we are doing a deep dive into this today. We're looking at a newly published 2026 paper from Cancer Cell by Entrialgo and colleagues.
It's a fantastic paper.
It really is. And the core of this research is about a revolutionary new class of drugs that finally figured out how to grab onto that smooth sphere.
Right, the impossible target.
Yeah. So our mission today is to unpack the sheer ingenuity of how these drugs work, look at the jaw-dropping clinical evidence in human patients, including that 44-year-old man we just mentioned, and then explore the terrifyingly smart ways cancer tries to survive anyway.
It really is a fascinating cat-and-mouse game at the molecular level.
Okay, so let's unpack this.
Yeah.
How do you stop a cancer cell that is permanently stuck in the on position?
Well, to appreciate the solution here, we really need to understand why that KRAS protein is such a nightmare when it mutates. In a healthy cell, KRAS is essentially a mechanical switch that controls cell division. It's not meant to be on all the time. It cycles.
Like a light switch.
Exactly. When it binds to a molecule called GDP, the switch is in the off position. The cell is resting.
Right, just hanging out, doing its normal cellular duties.
Right. Then, when the body actually needs new cells, it swaps that GDP for a different molecule called GTP.
So GDP is off, GTP is on.
Yes. The switch flips on, a signal is sent down into the nucleus, the cell divides, and then, crucially, the KRAS protein has an internal timer.
Oh, so it shuts itself off.
It does. It cleaves that GTP molecule, turns itself back into the GDP off state, and stops the division. But in cholangiocarcinoma, which, is a rising and incredibly stubborn gastrointestinal cancer.
Yeah, but 25% of these patients have a mutation, right?
Exactly. A mutation that physically breaks that internal timer.
The switch gets flipped to GDP, it turns on, but it can never turn itself off.
Yes. It is permanently jammed. The cell gets a relentless, endless signal to just keep dividing and growing. And the variations of this mutation are like a twisted alphabet soup.
Right. I've seen the letters.
Yeah. The most common is G12D, but there is G12V, G12A, Q61. They all result in that same catastrophic runaway growth.
Hang on. I need to stop you there because I read the medical news and I know we've actually had a major breakthrough with KRAS drugs a few years ago. Drugs like sotorasib, I think.
Yes, sotorasib and adagrasib.
Right. So if the switch is stuck on, why can't we just use the KRAS inhibitors we already invented to turn it off?
Yeah.
Don't those do exactly what we're talking about?
That is the logical assumption, and it's a really common point of confusion.
Mm-hmm.
But here's the mechanical catch with those first-generation drugs. They don't actually turn the switch off.
Wait, they don't?
No. They can only bind to the KRAS protein when it happens to naturally cycle into the inactive GDP-bound off state.
Oh, I see.
They essentially wait for the switch to turn off, and then they act like superglue, locking it down so it can't turn back on.
But wait, if the mutant cancer switch is permanently jammed in the on state, it's almost never in the off position for the drug to glue it down.
Precisely. It's a huge limitation. And there's an even bigger problem. Those older drugs only fit the exact physical shape of one very specific mutation called G12C.
Like a highly specific key.
Exactly. If you look at cancers outside of the lung, like our bile duct cancer here, the G12C mutation only accounts for about 1 to 7% of cases.
Wow, only 1 to 7%.
Right. So for the vast majority of gastrointestinal cancer patients, those older drugs are completely useless. The door is the wrong shape, and it's already open anyway.
That is wild. So you have a drug that works brilliantly, but only for a tiny fraction of patients.
Yeah.
Which brings us to the new breakthrough in this paper, RMC 7977 and its clinical human-grade counterpart, daraxonrasib.
Yes, the next generation.
The paper refers to these as RAS on multi-selective inhibitors. And they are fundamentally different because they are what biochemists call tri-complex non-covalent inhibitors.
Right.
Let's break that down. What does that actually mean for the drug's ability to grab that smooth sphere?
Well, it means they don't wait for the switch to turn off. These new molecules are designed to directly target the active GTP-bound on state.
Okay, but how? If it's a smooth sphere.
To solve the smooth sphere problem, they don't just try to stick to the KRAS protein alone, they bring a partner.
A partner.
Yeah. The drug binds to a highly abundant chaperone protein that is naturally floating around inside the cell called cyclophilin A.
Cyclophilin A. Got it.
When the drug and the chaperone protein link up together, they form a massive complex structure that's the tri-complex part that creates its own groove.
Oh, that is incredibly clever.
It forcefully wedges itself against the active KRAS protein and neutralizes the jammed switch.
It's using the cell's own proteins against it to build a custom clamp. That is brilliant. And the multi-selective part of the name, that implies it's not just hunting for that rare G12C mutation anymore, right?
Exactly. Because of how it wedges itself in, it doesn't need the exact lock-and-key fit of the older drugs. It can bind to a huge variety of these mutant forms, G12D, G12V, G12A, and it even hits different isoforms of the protein entirely, like HRAS and NRAS. It's basically a master key for the jammed switch.
And the data they present on just how targeted this is blew my mind. They tested this drug by measuring something called the IC50.
Right, a classic biochemistry metric.
Which, for anyone who hasn't spent time in a lab, is just a measurement of concentration. It's how much of the drug you need to drop into the petri dish to kill off 50% of the cancer cells.
Yeah, so a lower number means a much more potent drug. You want to use as little drug as possible to get the job done.
Right, so they test this new drug on mutant KRAS cancer cells, and the average IC50 was just 19 nanomolar.
19, tiny drop.
Exactly. Then they tested it on perfectly healthy wild-type cells. To get the same effect, they had to increase the concentration to 8,438 nanomolar.
Yeah.
That is almost a 50-fold difference.
And that number is everything in oncology. We call that the therapeutic window. It means the drug is essentially a sniper rifle for the mutant cancer cells, but largely harmless to normal, healthy tissue. It starves the tumor without carpet bombing the patient.
Okay, so let me do a quick mini summary here. Mutant KRAS is a broken on switch, right? And these new RAS-on drugs uniquely bind and trap this active switch across multiple mutation types by building a molecular wedge.
That's a perfect way to visualize it.
So for you listening, imagine a factory where the machines just won't turn off. If you can't hit the power button, how else would you halt production? You jam the gears. Keep that in mind as we look at the drug in action.
Because, as any scientist will tell you, a drug that works beautifully in a plastic dish doesn't always work in a complex, living body.
Right. Curing plastic dishes is easy. We need living models.
Exactly. So the researchers had to prove this in vivo. They moved to a really rigorous pipeline of mouse models.
They use CDX and PDX models, right?
Yeah. Cell-derived xenografts, where you grow human cancer cells in a mouse. And patient-derived xenografts, or PDX, which is even closer to reality, they take actual tumor tissue surgically removed from human patients and implant it.
And crucially, they used immunocompetent models, meaning mice that still have fully functioning immune systems.
Right, like the KP-TEACH and IAKP models mentioned in the paper.
So what actually happens inside the tumor when you give these living mice the drug? How is the cancer dying?
Well, the drug triggers profound apoptosis, which is the biological term for programmed cell death.
The cell's self-destruct button.
Exactly. A healthy cell presses it if it becomes damaged, but cancer cells usually disable it. This drug forces the cancer to press that button.
And they could actually measure this, right?
Yes, by tracking a biomarker called cleaved caspase-3. Think of cleaved caspase-3 like the debris left behind when a building is imploded. As the drug hit the tumors, the levels of this marker skyrocketed.
Wow, so the cancer cells were actively tearing themselves apart from the inside.
Right. And they were tracking the internal plumbing of the cell, too. These downstream signaling pathways with names like MEK-ERK and PI3K-AKT.
Let's clarify those. MECARC and PI3K-AKT.
Yeah, if KRAS is the main water valve at the top of the cell, those pathways are the massive pipes carrying the divide and conquer signal down to the DNA.
And what did the drug do to the pipes?
The researchers found that those pipelines were completely dry. The signal was totally shut down. They even found a gene called DUSP6 that acts like a molecular thermometer.
A thermometer for the cancer signal.
Yeah. As the concentration of the drug went up in the tumor, the expression of that DUSP6 gene went down in perfect lockstep correlation.
Meaning the drug isn't just killing cells randomly, it is hitting the exact target it was designed to hit. But you mentioned they tested this in mice with fully functioning immune systems. And reading the paper, this is where the story takes a really weird turn.
It does. It presented a massive biological puzzle. When they treated these immunocompetent mice, they looked at the tumor microenvironment, the area immediately surrounding the cancer. Normally, these cholangiocarcinoma tumors are cold, meaning the immune system just ignores them. But after giving the RAS-on drug, they saw a massive influx of immune cells flooding the zone.
We're talking heavy hitters, too. CD8-positive killer T cells, CD4 helper T cells, natural killer cells.
Right.
The drug basically rolled out the red carpet and turned the tumor into a highly immune-permissive zone.
The troops had arrived, exactly.
So, wait, here's where it gets really interesting. My immediate thought reading this was, oh, this is a slam dunk. If the drug is bringing the body's entire immune army right to the doorstep of the tumor, you just pair it with immunotherapy.
That makes total sense.
Yeah, you give them an anti-PD-1 drug to take the brakes off the immune system, and those T cells should completely wipe the cancer out.
You would absolutely think so. It's the most logical next step. But when the researchers ran that exact combination, it failed.
Completely failed.
There was virtually zero added benefit from adding the immunotherapy.
Oh, how is that possible? The immune troops are literally standing right there.
They are. But they dug into why, and what they found is incredibly sinister. They discovered a profound defect in the tumor's antigen presentation machinery.
Specifically, things called MHC class 1 and class 2 molecules. Let's translate that for everyone.
Sure. Think of MHC molecules like an ID badge that every cell in your body has to wear on its surface.
Okay, an ID badge.
Right. As proteins are made inside the cell, little fragments of them get displayed on this ID badge. If a cell turns cancerous, the ID badge displays abnormal, mutated proteins.
And then the immune system's T cells patrol around, check the badges, see the mutant protein, and destroy the cell.
That's exactly how it should work. But in cholangiocarcinoma, the cancer figures out how to stop making the ID badges entirely.
Oh, man.
It just stops wearing them. It essentially puts on an invisibility cloak.
That's terrifying.
It is. So the RAS-on inhibitor successfully damages the tumor and creates an environment that calls the T cells in. The troops arrive, but once they get there, they're functionally blind.
Because there are no ID badges.
Right. They are bumping right into the cancer cells. But without those badges, they have no molecular target to attack. They just don't see the cancer.
This explains exactly why immunotherapy has historically failed for these patients. And it completely changes how doctors need to think about combining treatments moving forward.
Exactly.
Okay, we are about 5 minutes in, so let's do a quick bulleted recap of what we've covered so far.
Good idea.
Bullet 1. Cholangiocarcinoma with KRAS mutations is highly aggressive because the switch is jammed on. Bullet 2. These new RAS-on inhibitors block the active GTP-bound state by wedging into it. Bullet 3. They shrink tumors in living mice and recruit immune cells. And bullet 4. Immunotherapy fails to synergize because the tumor hides its antigens.
Perfect summary.
Right, so seeing the drug work in mice, seeing how it alters the immune landscape, that is all incredibly vital science. But at the end of the day, curing a mouse is very different from saving a human life.
It always is.
And what makes this paper so compelling is that we don't have to wait to see if it translates. They included early phase 1 clinical trial data for the human version of the drug, daraxonrasib.
Yes, trilumbar RMC 6236001. This brings us back to the reality of the clinic. The paper details a few specific cases that highlight just how unprecedented this is. Let's look at case 1.
Okay, walk us through it.
This is a 60-year-old woman. She has metastatic cholangiocarcinoma, and her specific mutation is KRAS-G12V.
G12V.
She had already been through heavy-duty systemic chemotherapy, and the cancer just kept growing. So they enrolled her in the trial and started her on daraxonrasib.
And the response was dramatic. By her 7th cycle of treatment, her scans showed a confirmed partial response. The physical size of her tumor shrank by almost 40%.
38.6% to be exact.
Wow. And she didn't just shrink it.
Yeah.
She maintained that response well past 24 cycles of treatment.
That kind of durability in a disease that is famous for being stubbornly resistant to treatment is remarkable. And then we have case 2, which brings us all the way back to the patient we met at the very beginning of this deep dive, the 44-year-old man.
Right, but his mutation was different. He had the KRAS G12A mutation.
Exactly.
He had failed neoadjuvant chemotherapy. His surgery was canceled because the cancer had spread to his peritoneum, the lining of his abdomen, and he even tried immunotherapy, which we now know why that failed. He was out of options.
But on daraxonrasib, he achieved a 30% reduction in his tumors. And that response held for over 6 months, giving him a progression-free survival of 10.5 months.
I really want to emphasize why seeing these two cases side by side in the paper is such a big deal. You have a G12V mutation in one patient and G12A mutation in another.
Completely different genetic spelling errors causing the cancer.
Right. But this single drug worked beautifully on both. That is the master key in action.
It is.
So just to summarize this section, Phase 1 clinical data shows daraxonrasib safely and effectively shrinks tumors in humans across different KRAS variants.
It proves that the multi-selective capability isn't just a theoretical concept in a petri dish. It actually works in human beings.
Right. So I'll throw a question to you listening. If you were a doctor with a tool that works on multiple variations of a genetic mutation, how would that change how you sequence a patient's DNA? What happens when the tumor mutates again?
And that is the exact question the researchers had to face next because, well, this is oncology.
Right, cancer adapts.
The agonizing reality of treating cancer is that it is not a static, unmoving target. The tumor shrank, the patients bought precious time. But eventually, in many of these cases, the disease progresses. The cancer comes back.
Because you apply this massive evolutionary pressure to the tumor, you are choking off its primary survival pathway, so it has to adapt or die.
Precisely. It is a highly dynamic evolutionary system. So the researchers had to ask the next logical question. How exactly is the tumor fighting back? What are its escape routes?
Right, how does it pick the lock?
To figure this out, they took the tumors that had grown resistant to the drug and performed deep transcriptomic and whole exome sequencing.
Meaning they read the cancer's entire genetic code and the real-time instruction manuals it was printing out to see what it changed to survive.
Yes. And what they discovered is a phenomenon known as oncogenic flux.
Oncogenic flux.
The cancer fundamentally rewires its own circuitry to force that vital RAS signaling pathway back open. And the sequencing revealed 3 main evolutionary escape routes that the tumors were using.
Okay, let's walk through how exactly a microscopic cell manages to outsmart a modern drug. Escape route number 1 is something called KRAS amplification. And reading how the cancer actually pulls this off is honestly like reading science fiction.
It really is. It is the ultimate brute force strategy. The drug is highly effective at blocking the mutant KRAS proteins. So the cancer cell realizes it doesn't have enough functional protein to survive its solution. It just starts manufacturing massive amounts of extra copies of the mutant KRAS gene to overwhelm the drug with sheer volume.
But it doesn't just do this normally, right? It builds these things called extrachromosomal DNA or exedNA and homogeneously staining regions.
Right. Normally your DNA is neatly packaged into chromosomes. The cancer cell literally tears up the rulebook, rips pieces of the mutant gene out, and builds these circular rogue genetic factories.
Wait, they just float freely outside of the chromosomes?
Exactly. Their only job is to furiously pump out more mutant KRAS protein until there is simply too much of it for the drug to block.
It builds temporary illegal sweatshops just to survive the drug. That is incredible.
It's pure survival.
Okay, so that's brute force. What about escape route number 2? The paper calls this MYC overexpression.
So if you think of the cell like a command chain, the RAS protein is the general sitting at the very top, shouting the order to divide. MYC is the soldier at the very bottom, deep inside the nucleus, actually pulling the levels on the DNA to make the division happen.
Okay, so RAS is top, MYC is bottom.
The drug neutralizes the general at the top. So the cancer simply bypasses the chain of command entirely. It massively amplifies the MYC protein at the bottom.
Oh, they see.
The signal originates downstream, rendering the blockade up top completely irrelevant.
Got it. The worker just stops listening to the boss and starts doing it themselves.
Right.
And then there is escape route number 3, upstream RTK or SHP2 activation.
This is essentially the exact opposite of the MYC strategy. SHP2 is a signaling node that sits above KRAS in the cellular pipeline.
Oh, so even higher than the general.
Exactly. Instead of bypassing the general, the cancer hyperactivates the systems above him. SHP2 acts like a massive pressure pump, forcing so much biological signal down the pipeline that it just blasts right through the drug's blockade.
It feels like we are talking about fluid dynamics here. Like imagine the cell's signaling pathway is a massive rushing river.
Okay, I like this analogy.
The new drug, our RAS-on inhibitor, is a massive concrete dam we build right in the middle of it. The water flow stops, and the tumor starts to dry up and die. But, water always finds a way.
It does. It has to keep flowing.
So the cancer's first escape route, the KRAS amplification with those rogue xcDNA factories, that's the water just building up so high and so heavy that it eventually spills violently right over the top of the dam.
Exactly. Overwhelming volume.
And the second route, the downstream MYC amplification. That is the cancer tapping into a hidden underground spring that floods the valley from behind the dam, bypassing the river entirely.
Perfect.
And that third route, the upstream SHP2 activation, is the river carving a totally new high-pressure stream around the side of the dam to keep the valley flooded.
The cancer relies absolutely entirely on keeping the signaling river flowing at all costs. It is completely addicted to this specific biological pathway.
Right, so we're about 10 minutes into our deep dive now, so let's do our second bulleted recap.
Let's do it.
Bullet 1. Clinical cases prove human efficacy for daraxonrasib across different mutations. Bullet 2. Cancer resists by boosting KRAS copies using xDNA. Bullet 3. It reactivates downstream MYC or upstream SHP2. Bullet 4. The tumor relies entirely on keeping the signaling pathway open at all costs.
And that biological addiction is the cancer's greatest weakness. Because if we know exactly how the water plans to escape, if we can predict the tumor's evolutionary escape routes, well, we can cut them off before they even happen.
Right. If you know the river's going to carve a new stream around the side of the dam, you don't just build the dam. You build a secondary wall right where you know the water will try to go.
Exactly.
And that realization leads to the final and maybe most important part of this paper, the combination therapy roadmap.
The researchers didn't just document how the cancer resists, they actually tested solutions to prevent it. First, they looked at the standard of care, the chemo drugs, gemcitabine, and cisplatin.
The usual frontline stuff.
Yeah. When they combined those with the new RAS-on inhibitor in the animal models, they saw profoundly delayed relapse and extended overall survival compared to giving either drug alone.
But they got way more elegant than just adding chemotherapy, right? They went after the specific escape routes.
They did. Remember that third route we talked about?
Oh.
The upstream SHP2 node that acts like a high-pressure pump?
Yeah.
The researchers decided to combine the RAS-on inhibitor with a totally separate experimental drug called an SHP2 inhibitor. It's called RMC-4550.
So hitting the dam and the side stream at the same time.
This is what we call a double-down strategy, or vertical inhibition. By hitting the active RAS protein and the upstream SHP2 node simultaneously, you are choking off the pathway from multiple sequential angles.
Wow.
You block the main switch, and you simultaneously destroy the backup generator that the cell uses to try and force it back on.
It's like a medieval siege. If you block the main gate of the fortress, the enemy's going to starve. But if they have secret tunnels out the back, they survive. If you block the main gate and collapse the secret tunnels on day one, the fortress has literally nowhere left to go.
That is exactly what they saw in the lab. In highly aggressive, intrinsically resistant cancer cell lines models with names like SNU-869 and KKU-213A, this dual blockade completely overcame the resistance.
It worked?
The combination therapy caused the tumors to shrink, survival extended massively, and crucially, they looked at the safety profile. The body weights of the mice remained stable.
Which means the double therapy wasn't so toxic that it was destroying their healthy tissues along with the cancer.
Right. It confirms our fundamental thesis. Because the cancer is so fiercely addicted to this one signaling pathway, you have to maximize the suppression of that pathway to overcome its ability to adapt.
You can't just give the cancer one obstacle.
No, you have to trap it in a therapeutic corner.
So just to quickly summarize this final piece. To prevent resistance, combining RAS-on inhibitors with chemotherapy or upstream SHP2 inhibitors cuts off the tumor's biological escape routes.
Precisely.
So for you listening, think about that siege. If you block the main gate and the secret tunnels simultaneously, how long can the fortress hold out? Could combination therapy be the key to turning terminal cancer into a manageable chronic condition?
Synthesizing everything we've looked at today, it paints a deeply optimistic picture for the future of oncology. For a very long time, being diagnosed with KRAS mutant cholangiocarcinoma felt like hitting a brick wall.
Yeah, The biology was just too stubborn, and the target was deemed physically impossible to drug.
But this paper fundamentally shifts that reality. It proves that these devastating cancers have a fatal flaw. They are fundamentally addicted to RAS signaling.
And because they are addicted to it, they are vulnerable to it.
Exactly. These new RAS on multi-selective inhibitors like daraxonrasib represent a massive generational leap forward. They take a target that medical textbooks called undruggable and turn it into something highly manageable.
Especially when doctors use our growing knowledge of cancer evolution to build these clever multi-drug combination therapies.
It is a profound triumph of biochemistry over a disease that has outsmarted us for decades.
It really is. And I want to leave you, our listener, with a lingering thought today. For 40 years, the active GTP-bound state of KRAS was that completely smooth sphere. There were no pockets, no grooves. We were told it was chemically impossible to build a drug that could latch onto it.
But human ingenuity didn't stop.
Right. We figured out how to use the cell's own chaperone proteins to build a tri-complex wedge that finally cracked the impossible problem. Now that we have proven we can do this, what other impossible undruggable proteins of the human body are suddenly within our therapeutic reach?
That's the million-dollar question.
Could this chemical mechanism be the exact blueprint we need for curing the rest of our untreatable diseases? Thank you for joining us on this deep dive into the microscopic front lines. Keep asking the big questions.